Hello and welcome to another Mr. Carter Science Special. In this video we'll be looking at cloning, and in particular the 4 techniques that we can use to create a clone. So in this image we've got some idea of what media and films tend to think a clone is. We've got from the Star Wars franchise, Clone Troopers, Mini-Me from the Austin Powers franchise, but the only image that actually contains a real clone is this one here, with Dolly the sheep, who was cloned in Edinburgh University in 1996. So what is a clone? Well, a clone is an individual that is genetically identical to its parent, and therefore cloning is an example of asexual reproduction. There are 4 ways of creating a clone. The oldest of these ways is plant cuttings. More recently, that has been improved to allow plant tissue culture techniques. We have cloning of animal embryos and adult cell cloning. So 4 techniques that we can utilize to make a clone. Now, usually individuals are chosen to be cloned because of their desirable characteristics. For example, you may have a plant which is resistant to disease and therefore you wish to clone it, or maybe a plant that produces a particularly high yield, or maybe perhaps a cow that produces a high yield of milk and you therefore wish to clone those animals. So the oldest form of cloning is known as cuttings, and this occurs with plants. Cuttings are a form of asexual reproduction or cloning, and it's been used by farmers for thousands of years. So to take a cutting, you first remove a stem or a part of a leaf, as you can see here, and you put the cutting into a growth medium, normally that contains some auxin. The plant cutting will then produce new roots and shoots and grow into a new plant, and this is because plant cells can differentiate at any point in their life. So the cells that are in the stem of this cutting can undifferentiate and then redifferentiate and specialize to become root cells, which will then grow into the roots. The new plant that's produced in this technique will be genetically identical to the parent plant. This technique of cuttings is utilised for making many fruit trees in orchards, so that you can have identical cloned trees within your orchard, and therefore hopefully you'll be producing identical fruits that you can sell in the supermarkets. A more recent technique is tissue culture. Tissue culture is a similar technique to plant cuttings, but it can be carried out using just a few cells. So we don't need an entire organ of a plant, we don't need the whole stem, just a few cells. And we place them in a growth medium containing various hormones, including auxin. Typically this growth medium is on an agar plate. The cells will grow into a mass of identical cells called a callus, and the callus is then exposed to a different set of hormones which stimulate the callus to grow roots and shoots and leaves, and these new plants will be clones of the original parent plant. So we can then take those calluses and then put them into seed trays like this and then grow them as we normally would into new identical cloned plants. There is an issue with tissue culture in that it has to be carried out in sterile conditions, because the agar containing the growth hormones can easily be contaminated by fungi or bacteria. Tissue culture is widely used in the agricultural industry, and many plants grown in commercial nurseries are produced by tissue culture. Also, tissue culture is used to preserve rare plant species. The third technique on our list is embryo cloning in animals, for example cows, as you can see before. Here we take an embryo that's been produced in the usual way, sperm cells fertilizing an egg cell producing a zygote, which will divide by mitosis to produce a small ball of cells called the embryo. We can then divide that embryo by removing one cell from the embryo, separating it, and that cell, because it will grow by mitosis, will produce a new ball of cells, a new embryo. And if we remove 2 or 3 cells from the embryo, then we will end up with 2 or 3 new identical embryos, which can be placed into surrogate mothers into their uteruses, and the mother will therefore become pregnant and give birth to identical cloned calves, which will hopefully have the desired characteristics that you're after. So to look at that in a little bit more detail. So embryo cloning can be used to produce many more top quality clones each year. The best females are given hormones to make them produce lots of eggs. These are then artificially fertilized inside the cow using sperm from the best bull. The embryos can then be removed and cloned. The cells from the embryo are removed and grown into new embryos, and the new embryos are implanted into the uterus of a surrogate mother, and these embryos will then grow into calves, cloned calves, which will be identical. In this way, the best animals can be grown in large numbers in a short space of time. So, the most famous type of cloning is probably the cloning that produced Dolly the sheep, and this is known as adult cell cloning. And this diagram in front of you explains how adult cell cloning can be carried out. This is a typical 6-mark question. So pause the video and have a look at that diagram and see if you can work out how you get 6 marks explaining how to create a clone of an adult sheep. So let's look at that process in a little bit more detail. First thing to identify is which sheep you want to clone. And this is the sheep we are going to try and clone. So let's label that, try to clone this sheep. This sheep here is going to be an egg cell donor. We are going to use the sheep with the black face to donate an egg cell. So we remove the egg cell from the donor sheep, and most importantly, we remove the nucleus. Take the nucleus away, so we end up with an empty egg cell. At exactly the same time, the sheep that we're trying to clone, the sheep with the white face, we remove a cell from the skin cell from around the udder, and we take the nucleus from that skin cell and we place the nucleus into the empty egg cell. So we end up with an egg cell now which has a diploid nucleus, a nucleus with a full set of chromosomes. If we then give that a small electric shock, that nucleus and the cell will begin to divide, and the zygote will turn into an embryo. But every single nucleus in the embryo will be genetically identical to the sheep with the white face where the nucleus came from originally. If we now take that embryo and we implant it into a third sheep known as the surrogate sheep, that sheep will become pregnant. The embryo will grow into a fetus and eventually into a lamb which the surrogate mother will give birth to. Now that eventual baby, the lamb, will have every single cell being genetically identical to the original sheep with the white face where we took the nucleus from. Now that's quite a complicated idea to grasp, so I'm going to go through it again on the next slide. So here is that same idea again, but this time with a little bit of writing. So adult cell cloning is a technique that created Dolly the sheep, and as we've already said, it's a form of asexual reproduction. So the first thing to do is to take an egg cell and remove it from a female sheep, also known as a ewe, and the nucleus is removed from this donor egg cell to create our empty egg cell. We discard that nucleus, we don't need it. Remember, that would only be a haploid nucleus, it is of no importance to us. But at the same time, a nucleus is taken from an adult sheep's skin cell from just under the udder, and that's taken from this sheep here with the white face. The nucleus from the adult cell is inserted into the empty egg cell to form a diploid zygote, and this cell is given a small electric shock which stimulates our zygote to divide by mitosis and to form our embryo. We then take that embryo and we implant it into the surrogate ewe's uterus, and the embryo will grow into a fetus and then a baby sheep. And that baby sheep, a lamb, will have the same genetic material as the adult sheep, which donated the skin cell and the nucleus. So that was the first technique. It was carried out in 1996 in Edinburgh University. So here you can see, here is Dolly the sheep. She was the very first cloned animal using the adult cell cloning technique, born in 1996 after a team of scientists from Edinburgh University produced her using an adult cell from another sheep. This one here, you can see that's the surrogate parent. Dolly, though, it must be said, was the only success from hundreds of attempts. So this is by no means an easy technique to employ. But it does have many advantages. Adult cell cloning will allow animals that have been genetically engineered to produce proteins in their milk to be produced in large numbers. So we can link this technique in with other scientific techniques. So if we have inserted genes into a particular organism, which is in itself, it has a low probability of success. But if we have been successful and we now want to produce thousands of clones of that individual, then we can use adult cell cloning to do that. This technique could also be used to save some animals from extinction. However, adult cell cloning has many disadvantages. As we've already said, the technique is often unreliable and it is very difficult to carry out. It could lead to the cloning of human babies, which of course would be highly unethical. And we also have a big issue that adult cell cloning will reduce variety in the population, and this makes cloned populations vulnerable to disease and environmental change. I hope you've enjoyed this video on cloning. If you have, don't forget to like and subscribe, and let me know in the comments what you have learned. Thank you very much.